Now we will take a little break and describe what our laboratory has contributed on NF-kappa B signaling. Long back in 2003 we had a paper. If you remember, I told you one of the figures from our publication was in fact incorporated into the textbook of cancer biology by Weinberg, a molecular biology of cancer by Weinberg. So, I will just tell you some of the details on that work. Before doing that, because that work was on cervical cancer, I need to give you some background on cervical cancer. As we already discussed, cervical cancer used to be the number one cancer in India among women in terms of incidence, but now it is taking the second place. But of course, in the worldwide, second most common cancer and most common cancer among Indian women, it used to be, but now it is also taking second place in India also. This is an old slide. So, about 4.7 lakhs new cases and 2.3 lakhs cancer death were reported in the globally. In India, about 1 lakh new cases, about 70,000 cancer deaths, which basically account for about 26% of the global incidence. This data I am just specifically giving for cervical cancer, because we are discussing cervical cancer. Now, what is important in cervical cancer is what causes it, right. So, human papilloma virus infection is the one that is said to be the major high risk factor that is causing this cervical cancer. A German scientist discovered that, and I am taking his quote, HPV infection is necessary but not sufficient. What he means by that is, more than 90% of the cervical cancer patients always had HPV infection, but does not mean that all those infected with HPV will get cervical cancer. I hope you understand that. In the patients, if they are already having cervical cancer, you will definitely find HPV, but just because you are infected with HPV does not mean you will get cervical cancer. So, that is what it means. Why? Because our immune system protects us and there are many other factors. So, ultimately the concept is HPV infection is necessary, but not sufficient. Other factors in your system, because each individual is different. So, how do you protect yourselves? It is a different thing. So, that is why although HPV infection is the major risk factor, molecular alterations of tumor suppressor genes or oncogenes either associated with HPV or even not associated with HPV, they are necessary for the progression of cervical cancer. The HPV infection necessarily initiates cervical cancer, but then for it to progress to the full level cancer, you need additional host factors. So, now you can understand NF-kappa B could be one of the factors, because just now I told you NF-kappa B expression, overexpression also helps. So, this is the background. Cervical cancer is usually classified as an epithelial tumor. 70% of the cancer patients belong to the first type, squamous cell carcinoma of the, you have in the cervix, you have the ecto layer and endo layer. So, ectocervix, about 70%, average I said 60 to 80%. HPV 16 is the usual risk factor. Whereas there is another type of cervical cancer called adenocarcinoma. Whenever you see adeno, the word adeno, breast also it is, we say that adenocarcinoma of the breast. Adeno means secretory. Usually adeno, that word refers to the cancers of the secretory type. So, here less than 20% or approximately 20 to 30% of cervical cancers belong to this category, and here HPV 18 is the risk factor. So, there are many HPV types, more than 80 types of HPV are known, and HPV 16 and 18 are called high risk HPV types. There are some more high risk HPV types, but these are the most common ones, because I do not want you to confuse with 80 different HPVs and then tell that. By the way, did you listen to that TGF beta lecture? Yeah, some of you came. So, you see I have given an introduction to TGF beta signaling as a tumor suppressive pathway, but it also has proliferative this one. I purposely did not tell for simplicity, because it is a dual action, I know about it, and similarly there is some tyrosine phosphorylation there, which we have not discussed, because it is mainly serine threonine phosphorylation that drives the TGF beta signal. Just I just told in between, just for you to remember. When I did my postdoc in Israel and then in US, and then when I came back to Trivandrum, I wanted to start something on my own. So, I just asked around what is the most prevalent cancer in India. The answer came, it is cervical cancer. At that time it was 1998, something like that. Yeah. So, then I went to some hospital there nearby and asked how the pathologists classify this. They have a immunohistochemistry grading of this. It was very interesting. The pathologists can distinguish, see, normal epithelium and then what is called low grade squamous intraepithelial lesion. They call it as LCIL. And then they can also identify, by looking at the microscope, they can identify based on the lesions, morphological changes that occur, high grade squamous intraepithelial lesions and then squamous cell carcinoma. So, this is how they have kind of graded them. This is not classification as such, it is grading, histological grading, because clinical classification is different. There they will say stage 1, stage 2, stage 3 and so on. So, do not get confused with that. And also the clinicians will look at the lymph node presence of certain abnormalities in the lymph node, and accordingly they will classify, and based on the tumor size also they will classify that. So, you should not be confusing with that. This is histological grading. And as I told you, there are number of papers at that time that showed that NF-kappa B is activated, constitutively activated in different cancers, none for cervical cancer, and it is important in India. So, I thought it is a good idea to check whether NF-kappa B is constitutively activated in cervical cancer. So, that was the basis, that was the logic. Why I am telling all this is because when you want to start a work, you should have some hypothesis, you must have some logic, and if possible some relevance to the local environment. So, this is how we started as someone who has just been trained on that and came back, I started this. First I took the help of pathologists with some clinical samples. As I told you, they have classified, they have graded them as LCIL, low grade, high grade and squamous cell carcinoma. So, we compared the expression of classical NF-kappa B. You remember classical NF-kappa B means P50 and P65 or Rel-A. P65 is also called Rel-A. So, P50, Rel-A and its inhibitor I-kappa B alpha. So, we stained the samples available to us with all these factors. We stained for P50, Rel-A and I-kappa B alpha. What is very interesting about this, incidentally this is the figure that was taken in the book also, especially the I-kappa B. I think the next one, that is there is one more, yes, this figure I-kappa B phosphorylation was what was taken. So, we will come to that. So, the staining showed the P50 Rel-A in the normal sample mostly in the cytoplasm. I kappa B also, staining is seen in the cytoplasm. Whereas, in the low grade and especially in the high grade, you can see here, it is the nucleus is also heavily stained, REL A and P50. So, what it means is, as the cancer progresses with reference to histological grade, these factors have moved to the nucleus. That is what you call as activation. So, both P50 and REL A moved to the nucleus. At the same time, you remember the signaling pathway. I kappa B gets degraded when NF kappa B gets activated. So, you see a degradation here. I kappa B seems to disappear as the cancer progresses. In the squamous cell carcinoma, this blue color indicates that it is completely absent. Only the brown color indicates its staining. So, you can see that it is getting degraded. So, as expected, the NF kappa B is constitutively activated. So, the factors P50 and REL A, they move to the nucleus, and then simultaneously I kappa B gets degraded. So, this is what we can conclude from this figure. Of course, this one experiment is not enough, people will not accept. In fact, even after we published, somebody published a little bit different from this. They said only P50 is moving to the nucleus, but not P65, something like that. There were some controversial publications, but then we had enough data and we published in a very good journal called Oncogene. We have also analyzed this data with so many samples. You can see about 18 here, 23 low grade, 27, and again in the squamous cell carcinoma, the pathologists have classified as well differentiated squamous cell carcinoma, moderately differentiated and poorly differentiated. Which one is more aggressive? Poorly differentiated, because cancer cells are poorly differentiated. So, in order to know that, we have also looked at that. You can see that as we go more aggressive, the more translocation to the nucleus, that is 81%, 92%, you see that P50 and REL A, and almost nil for I kappa B. So, this kind of gives us a complete clue that it is constitutively activated. In the tissue, there is a problem, you cannot do any mechanistic studies, right. You cannot give something, induce something and all, because it is a tissue. So, we can only do immunohistochemistry, we can do some other assays also. As we go along, I will tell you. We even took some of the tissues and did Western blotting. Here also we got the same results, that is, we have fractionated the nucleus and cytosolic fraction. We had the nuclear fraction and the cytosolic fraction. As you can see, N means normal, C means cancer. So, in the cancer samples, you see the P50 and REL A being in the nucleus, but cytosol also has obviously, but relatively more is there. So, indicating the translocation that we postulated based on the immunohistochemical data. Similarly, I kappa B alpha also, while you find that in the normal tissue, you do not find that in the cancer tissue. We have done with several, beta actin served as a control. I am just telling how we people do experiments, and also how the same findings are confirmed by different approaches, so that you will be able to make an authentic statement. Otherwise, people will not believe. Just you do immunohistochemistry and only and then show, then people might say that it is qualitative, it is only just visual, it may be wrong, maybe it is a technical problem, how did you do it with so many samples, statistically is it significant, all kind of questions will come. So, if we confirm that by Western blotting, and then by one more, I will tell you what it is. We looked at by this is RT-PCR also we have done. So, we looked at the expression. So, the expression of I kappa B alpha increases, that is there in cancer also, but ultimately this is at the mRNA level. Here also there is another important point. At the mRNA level you find more of I kappa B alpha, but at the protein level there is nothing. So, it gets degraded. More and more is synthesized, but then it gets degraded at the protein level. So, that is also shown here. And then this is a very important assay which almost confirms the binding of NF kappa B in the with the DNA, because you see what is the use of showing translocation. Okay, I can argue that the transcription factor translocates to the nucleus, but does not bind DNA. So, you have to demonstrate nuclear binding. How do you do that? How do you demonstrate a protein binding to the DNA? There is one simple assay, which is what is this assay? Anybody familiar with this? Huh? No. This one is, this is not that. This is gel shift assay. Okay. So, this is a simple assay which tells you, you take a radioactive, you know where NF kappa B binds. It is a NF kappa B binding site, it is an oligonucleotide. You can label that with P32 radioactivity. Once you do that, now you put the nuclear extract which is supposed to have the P50 and RelA, it is supposed to bind. So, you add that, incubate that with this radioactive labeled oligo, which contains the binding site. Control will be some other oligo which does not bind, that can be a control, or alternatively we have several controls, but I will just tell you. The other control can be the unlabeled oligo can be added to this mixture. So, what will happen? The unlabeled will replace the radioactivity. So, you will not see the radioactive band if it is correctly binding. So, these are some of the controls. Now, once you run it on a gel, usually native gel, because we do not want to separate the proteins. So, when you run it, what will happen? The free oligo, labeled oligo, which is not bound, will move faster, right. In the sense they will be at the bottom. Those which are bound with the protein, they will have a high molecular weight, because protein is a big molecule, oligonucleotide is small. So, naturally it will come to the bottom, but if it is bound, then this is often called gel retardation assay also, it is called. It is also called EMSA, electrophoretic mobility shift assay, it is popularly known as EMSA. ELISA is different, this is EMSA, electrophoretic mobility shift assay. So, that way it is very popular. This is one of the assays that people do for knowing if a protein binds to DNA, specific sequence. So, that it is. There is another control that we usually do in this. We give an antibody to RelA and P50. Now, it will not be able to bind, because the antibody will compete to the site, and so it will not be able to bind. So, it will displace that band, and the band will be shifted also. So, these are the different things we do, and thereby we demonstrate that constitutive NF kappa B DNA binding activity happens during cervical cancer progression, because we have used a normal, low grade, high grade and cancer, and demonstrated that there is indeed NF kappa B DNA binding. So, this is an important experiment. Now, if what we say is true, I kappa B gets degraded when it is getting phosphorylated. So, we have to demonstrate whether that I kappa B was really phosphorylated. We took a phospho I kappa B alpha antibody, and so we demonstrated that. You see here well differentiated squamous cell carcinoma, moderately differentiated squamous cell carcinoma and poorly differentiated, and this low grade, high grade. You see here in the normal you do not see much, but you see a very high phosphorylated I kappa B even in the low grade. As you go along in high grade you see more of it, but when you come to squamous cell carcinoma you do not even see the phosphorylated I kappa B, because the whole I kappa B is degraded now. So, this clearly demonstrates the sequence of events, that is I kappa B is getting phosphorylated and then finally it is getting degraded also. So, this clearly demonstrates the constitutive activation of NF kappa B during cervical cancer progression. This is what we finally published in Oncogene. There was another interesting finding. You remember I told you there are two types of cervical cancer, squamous cell carcinoma and then adenocarcinoma. Adenocarcinoma constitutes only about 30%. So, getting samples was actually very difficult. We were in Trivandrum at that time. There is a regional cancer center there. Those people have given us samples, but we could not get to this adenocarcinoma. Some of it I think we obtained that, and we did some more experiments with some samples obtained from Punjab also. There is one postgraduate Institute of Medical Education and Research, PGI as they call. So, from Chandigarh also we got some samples. It is very interesting here. I am going to tell you something that we initially could not unravel what and why it happens. What happened here is, normally in cancer you will find that I kappa B gets degraded and P50 and REL-A will go into the nucleus, that is what you will find. But this immunohistochemistry with adenocarcinoma, we found even in cancer there is so much of I kappa B. So, now we got confused whether adenocarcinoma, whether it is constitutively active, because why there is so much I kappa B. Here we tried to sequence that I kappa B and tried to see whether there is any mutation because of which it is there, nothing. In the literature also I told you in solid cancers there is no mutation of I kappa B alpha, and we looked at, we sequenced this I kappa B and we saw it was only wild type. So, we were at a loss. That paper we published, squamous cell carcinoma we published, this was an extension. So, I just told the student, hey, we have done all the work only in squamous cell carcinoma, why not we do something with adenocarcinoma also. So, this was a very surprising finding to us, and we were at a loss how we can say that NF kappa B will be constitutively activated when there is so much of I kappa B, because I kappa B is a known inhibitor. So, this was the puzzle, and we tried to do the same thing with blotting also. We confirmed, first we thought it may be a technical error with immunohistochemistry, no, we again found the same results with western blotting also. Again we are finding I kappa B in the nucleus also. See, nuclear fraction, again you are finding it here also in the cancer samples. In the normal samples it is there, but very little, but in cancer you are finding it. Cytosol you see in the normal there is so much, that is expected, but in cancer you do not expect it, I kappa B, it should get degraded. So, we were puzzled by this and looked at the literature long back in pancreatic adenocarcinoma or some, I think pancreatic adenocarcinoma, also some people found I kappa B being expressed, but surprisingly no explanation as to why it is there and how to explain that, and when there is no mutation. So, we have shown the DNA binding activity again by in adenocarcinoma also. So, DNA binding activity is there, how come when there is so much I kappa B? This is a puzzle. Actually, this work we have published, I told you NF kappa B is constitutively activated. In this one we have not said anything about adenocarcinoma, because at that time we did not do. It was something after we published this. This is what we have done. And so far we did not get a very clear clue for that. What I read from the literature is that if REL A is acetylated, then I kappa B alpha, even if it is present, it may not be able to bind that. That is what we came to know. So, we blotted with acetylation specific REL A antibody and found that it is staining with this REL A in the cancer adenocarcinoma. So, this was the kind of conclusion, but then we did not publish that yet. So, we think probably the REL A gets modified in such a way that it is not able to bind I kappa B, but I kappa B is present. This is a very novel kind of explanation, but that has not been we have not been able to publish because the quality of the acetylated antibody was not very good. So, some reviewers have rejected it, and the student also left the lab and we could not do that. So, we had a number of publications on NF kappa B. For those of you who are interested in research, can look at that. We have shown that I kappa B and some chemical inhibitors of NF kappa B can sensitize the cells to cisplatin. This is another work. That is, you give cisplatin, you give some NF kappa B inhibitor, for example, curcumin, the cells are dying better. So, we have shown that, and we have also published a little bit other molecular. We have written a review also on this with Bharat and so on. This came in that some Nature News India magazine also. So, we will kind of, as I just told you, this is also finding a part of this book. This is the book Biology of Cancer by Bob Weinberg. We will kind of stop here, but if you have questions we can discuss that.